Hello and welcome to Virtual Investor Conferences. My name is Greg Young, and on behalf of OTC Markets, as well as our co-host Zach, Small Cap Research, we're very pleased you have joined us for our next live presentation from NeuroBo Pharmaceuticals. Please note you can submit questions for the presenter in the box to the left of the slides. You can also view the company's availability for one-on-one meetings through the Scheduled Meetings tab found on the conference platform. At this point, I'm very pleased to welcome H.H. Kim, Chief Executive Officer and President, and Marshall Woodworth, Chief Financial Officer of NeuroBo Pharmaceuticals, which trades on the Nasdaq under the symbol NRBO. Welcome, H.H.
Thank you, Greg. Hello, everyone. Thank you for attending this conference and sparing your 30 minutes with us. I'm H.H. Kim, CEO and President of NeuroBo Pharmaceuticals. We're listed on Nasdaq with the ticker symbol of NRBO. Now, quickly looking at our leadership team, myself, we have Marshall Woodworth, the CFO, with me today. He has 35 plus years in financial industry. And we have the non-executive management, Mi-Kyung Kim. She's the Chief Scientific Officer, 25 plus years in drug discovery. She's currently the head of research at our largest shareholder, who is Dong-A ST in Korea. We have Dr. Chris Fang. He's the Advisor Consulting Chief Medical Officer with 20 plus years in clinical development. And we have Robert Homoka, SVP Clinical Operations with 35 plus years in drug development. Now, he is the one who's leading the charge with our two clinical programs. Now, who are we? NeuroBo Pharmaceutical, we're targeting obesity MASH, so basically in the metabolic space. We have pipeline of next generation therapeutics. First asset, DA1726. This is the GLP-1/glucagon dual agonist. We have ongoing phase 1 trial for the treatment of obesity. We had the part 1, which is the SAD study for the phase 1. Interim top-line data results came out, and that was for the planned cohorts, and it showed strong safety profile. So we are adding additional cohorts to the SAD study and the MAD study to see and explore what is the maximum tolerable dose. Now, part 2, which is a MAD study, 4-week study for this drug, we already started it during the SAD study because we believe that whatever we've been seeing in the SAD was very comfortable with the safety profile and the tolerability to start the 4-week MAD. So we're on the MAD study as we speak, and we're hoping to have the interim data readout for the planned cohorts during the first quarter of next year. Now, after we released the SAD part 1, the interim data readout, we did see a little bump down of our share prices, and I think my guess is that the market wanted to see some kind of a weight loss data from the SAD study, but have to remember, SAD study is a single dose, and super majority, almost everyone in this field, they always release SAD and MAD together. The only reason we released the SAD top-line data, the interim data, was because we wanted to make sure that we keep the promise that we are transparent with our shareholders and the investors, and that we are cruising through the SAD and MAD. That was the reason for that press release. It has nothing to do with the safety or any other weight loss data that we've seen. Now, with all that, we are running additional cohorts in the SAD, and at the same time, we are still running the MAD study as well, and we're hoping to have that data readout during the first quarter of next year. The second asset that we are focusing on is DA1241. It's a small molecule GPR119 agonist, and we have an ongoing phase 2A with subjects with presumed MASH. We had the last patient out the other week, and we are in the process of closing the study. With all that, we are hoping to see the top-line data readout during some part of next month. Now, we are backed by a strategic partner and major shareholder of Dong ST. This is a Korean pharmaceutical company, has been in business for almost 90 years in Korea. Unlike other biotech companies, these two assets came from Dong, and they are backing us up with providing half of the financial raise we have done in the past for 2 rounds. And because of this strong partnership between Dong, we are able to move forward to see how these drugs work in humans. We're well capitalized with approximately $21.7 million in cash at the end of Q3 of this year. Now, looking at multiple near-term milestones, we'll start with the 1241, the MASH drug. We had the last patient last visit during the last quarter, and we are hoping to have the top-line results out by Q4, which probably will be next month. After that, in 2025, we'll be having meetings with the FDA and seek what will be the next steps. Now, for DA-1726, the obesity drug, now during the third quarter this year, we had the interim results released on the SAD study. Now, after that, we are running with the SAD, more cohorts in the SAD to see the maximum titratable dose. And while we're on it, we're working on the MAD study, the part 2 as well. And in the first quarter of 2025, we're hoping to have the last patient last visit and have the interim results of the MAD study out during the first quarter of next year. Once we have all those in-human data, we are planning to start a next step, part 3 of the phase 1, which will be a 24-week study. But depending on how we see the drug worked in human, there could be other options to take as well. Now, DA-1726, this is the obesity drug. It is a GLP-1 receptor glucagon receptor dual agonist. This slide and the next slide will be the most important slides in the 1726. We'll start with the competitive differentiation slide. This is a whole table where GLP-1 glucagon assets in clinical are shown. Now, when we start from the left side, pemvidutide developer is Altimmune. It's phase 3 ready, and it's very well balanced as Altimmune announced. GLP-1 receptor, glucagon receptor being 1-to-1 in its potency ratio. With the 1-to-1 very balanced approach, body weight loss of 15.6% was seen at 48 weeks. Now, what are we? 1726, same GLP-1 glucagon dual agonist. Our balance is 3-to-1. Now, look at mazdutide, the next one. Developer is Innovent Biologics. Eli Lilly licensed it in. They have the NDA in China for 6 milligrams, but Eli Lilly is doing a phase 2 with 16 milligrams in the US. Although it's not disclosed on how the balance works, within our research center who did all these balancing analysis, it did show to be about 6-to-1. Once weekly injection with 6-to-1 balance, 18.6% at 48 weeks. This is for the 9 milligrams. For the 16 milligrams, it's about 23% weight loss. Survodutide, Boehringer Ingelheim, Zealander, currently in phase 3. The efficacy balance between GLP-1 glucagon is 8-1. That was released by Boehringer Ingelheim. Although when we did it in our research center, it showed about 5-1. So with 8-1 balance, body weight loss of 18.7% at 46 weeks. Now we have semaglutide and tirzepatide on the right end, but just looking at all these different balancing acts between these two agonists, the results are quite clear that the balance does have effect. So mazdutide 6-1, survodutide 8-1. When you look at the body weight loss, 9 milligram, high dose of survodutide, all of them tends to be around the 18% range, while 1-1 does show a little lesser weight loss of 15.6. Why are we 3-1? Our research center, when they were looking into 1726, they wanted to make sure that GLP-1 and glucagon, both of the efficacy shine. But when you look at Altimmune, although it's 1-1, because of being 1-1 well balanced, it kind of balances out the GLP-1 effect and the glucagon effect, meaning that they do not have the glucose lowering effect coming from the GLP-1. We didn't want that. We wanted to make sure that there is that glucagon lowering effect, while we make sure that the glucagon has a glucose lowering effect, while from the GLP-1, while we have the glucagon effect shine as well. And that is the reason why we went with the 3-1 approach. Now, these are the key attributes that we're looking for in human clinical trials. These are based on our known clinical studies, and we're hoping to see these blossom in the clinic. The first one, it would be, this is a obesity trial and weight loss drug, so change in body weight. We're hoping to see similar or better than comparison. When we did a head-to-head non-clinical study with survodutide, we did see about 7% more weight loss compared to survodutide. Between semaglutide and 1726, we saw about 8% more weight loss while consuming 8% more calories. And compared to tirzepatide, head-to-head, similar body weight loss while consuming 20% more calories. Basically meaning the tolerability was much better and the side effects were much well controlled in these non-clinical studies compared to semaglutide and tirzepatide. Tolerability, compliance, we hope to see similar or better. Same here, survodutide, 7% more weight loss, but semaglutide, tirzepatide, as I mentioned, the mice on these drugs, compared to mice on these drugs, 1726 induced mice ate a lot more food while losing more weight. Glucose control, insulin sensitivities, we're hoping to see similar or better than competition. Head-to-head with survodutide, 1726 effectively lowered TCHO and TG and glucose level. Head-to-head with semaglutide had better HbA1c in glycemic control. And compared to tirzepatide, had better glucose lowering. But this wasn't a type 2 diabetes-induced mice model. It was more of the obese mice, but still had better glucose lowering effect. Body composition, meaning fat lean mass loss, we're hoping to see better than competition. We'll show it to you in the later slides. Compared to survodutide, 1726 demonstrated superior body fat mass reduction and have relative lean body mass preservation. With semaglutide, 1726 had better expression of thermogenic genes in white adipose tissues, and white adipose tissues is the target to lose more fat. And we haven't done this type of study compared with tirzepatide yet. On MASH, NAFLD, better than competition. Haven't done a direct head-to-head with survodutide, but with semaglutide, 1726 had better NAFLD activity score and fibrosis resolution. Now, with all that, there are a couple of other key attributes that could lead 1726 into best in class, which will be weight loss matrix, cardiovascular, but all these will come through as we progress through the clinical trials. Mechanism of action. GLP-1 receptor will reduce food intake while glucagon will increase energy expenditure. Now, this is a comparison with the survodutide in weight loss and lipid lowering. When you look at the weight loss side, have about 17 on the left side, 1726 induced mice lost about 7% more weight loss, while 1726 induced mice food intake was a little higher than survodutide. But when you look at the biochemistry, TCHO, TG, and glucose lowering effect was quite higher than survodutide-induced mice. And thermogenesis-related gene and BAT, you can clearly see that 1726 had more showing compared to survodutide. Now, continuing with the survodutide comparison, when you look in the middle, 1726 induced mice, when you look at the W2 on the right circle, you can clearly see lean body mass was increased by 8%, while all of that came from loss of body weight. So body fat from 41.3% was reduced to 33.6%, meaning all the weight loss was mostly coming from the body fat loss. Below that is survodutide. When you look at the body fat mass change, it only changed about 2%. So clearly, 1726 has more effect on the white adipose tissues. When you look on the right, HF control, 1726 and survodutide, you can clearly see 1726 has more effect into the white adipose tissue, and it's breaking it down better than survodutide in non-clinical settings. With that, just a quick look at potential in MASH. This is a head-to-head comparison with semaglutide. On the left, it's the steatosis and inflammation. You can clearly see that the semaglutide on the top right corner, compare that with the 1726, much better control over the steatosis and inflammation. And on the middle, you can see the fibrosis. On the top right, semaglutide still having a lot of the white points, which means it's more fibrosis still left there. But the 1726 high dose clears the fibrosis a lot more than semaglutide. And even the NAFO score, when you look at semaglutide being on the 5, while high dose of the 1726 going down to 3.6. And another slide on this, it's quite interesting when you look in the middle. You can clearly see that glucose lowering effect is much better by 1726 than a semaglutide. And of course, the body weight loss on the left is clearly better. Now, what are we doing at the moment? As I mentioned in the front, phase 1, we have part 1 and part 2 ongoing. Part 1 is the SAD, single ascending dose study. Part 2 is the multiple ascending dose study. We have both of them running at the same time. For the SAD, we did go over what was planned to make sure that we go up to as far as we can to see how far the tolerability and the safety issues can be seen. And the higher we get, we're hoping to see much more weight loss. That is the goal for us. We're running the part 1 and part 2, and we're hoping to see the MAD study, which is the part 2 interim data readout during the first quarter of next year. Once we collect all those data, we are thinking about the 24-week part 3 of the phase 1 study. This will explore maximum titratable dose and dietary changes, plus including BMI and a lot of different data sets to show early sign of efficacy. But depending on what we see in SAD/MAD, we may go to a different route of starting a phase 2 rather than this 24-week study. But everything currently depends on what we see during the SAD and MAD and the characteristics of 1726. Now, 1241, this is the oral small molecule potential for best-in-class GPR 119 agonist for treatment of MASH. 1241, as a mechanism of action, it generates more GLP-1s and natural GLP-1s in the gut, and from then on has a lot of effects into the liver and pancreas. And we're hoping to see that it has a superior anti-inflammatory effect in MASH and in the liver. That is our focus point. And we are running the phase 2 with 1241 standalone and 1241 with DPP-4 inhibitor combination. The reason we picked DPP-4 is because of its mechanism of action of prolonging natural GLP-1s in the gut. So once we have these data out, we did not do the biopsy during the phase 2A because this is more focused on anti-inflammation. We already seen that 1241 is very safe during our phase 1 in the US. So very safe, orally available, anti-inflammatory focused drug, we believe will have a good positioning within the MASH field, plus in other areas as well. Now, can anti-inflammatory action be the main focus of MASH? Probably not, because MASH is very complicated disease where inflammation, fibrosis, steatosis, all of these things come into action, even glucose control. So what we are planning to do is once we have the first in human phase 2A results out, we will be looking into different combination therapies, such as a combination with Madrigal's resmetirom, semaglutide, or FGF21s. And one of the reasons for that is they're more focused, like at least semaglutide and FGF21s have more focus as fibrosis resolution compared to our drug. And there could be a very big synergistic effect once administered as a combination. So, phase 2A, we have part 1, which is a standalone, and part 2 with sitagliptin, the DPP-4 combination. Number of subjects is approximately 90 subjects with presumed MASH. We have about 25 sites in the US, and we're hoping to have the top-line results during next month. Now, going into financials, as of September 30th, the end of third quarter, we have $21.7 million in cash. We do have 2 outstanding warrants from our past financing, Series A warrants. The strike price is $3.93, and the trigger point will be a successful MAD study from 1726. And if and only if all of those warrants are exercised for the Series A, we will have about $20 million coming into the company. Series B, it's a successful part 3 of 1726. That is about 30 million additional cash that may come in if fully exercised at the price of $3.93. Again, just to wrap up, we have 2 assets, metabolic assets in clinical, DA1241, the GPR 119 agonist, small molecule orally available. We're looking at top-line results during next month. 1726, the GLP-1 glucagon dual agonist, currently in SAD and MAD, and we're hoping to have the MAD interim data readout during the first quarter of next year. So I think that's where we wrap up. Metabolic focused company, NeuroBo. Remember that we do have an obesity drug in place. We are sailing through the SAD and MAD, and the only reason we're pulling up the cohorts is to see where it can go. More efficacy or higher dose, or if it's tolerable, we may be able to see more weight loss. That's the reason why we're doing it. Thank you.
Thank you. We have a number of questions. I'll start with the top of the list and read down. First question, can you elaborate on how DA 1726, your obesity drug, operates and how this is different than the competitors?
Operation, I guess it's a mechanism of action, right? GLP-1, semaglutide is GLP-1. I think everyone knows how that works. You're inducing GLP-1 hormone into the human body. Tirzepatide is GLP-1 plus GIP, and they're injected into the body. And GIP actually enhances and effectuates the GLP-1 effect. Ours is GLP-1 plus glucagon. So the approved two drugs, they're very focused on GLP-1 and the mechanism of action. And GIP just helping it enhance the GLP-1. Now, ours is dual. We have the GLP-1 effect plus another mechanism of glucagon. Now, GLP-1, everyone knows, reduces food intake, lowers glucose. Now, glucagon, what it does is that it burns energy, basically burns energy. And we're hoping that GLP-1 will reduce the food intake and lower the glucose level, while glucagon will burn the energy to make sure that there is a fat burn. And that's one of the reasons why when you look at pemvidutide, survodutide, mazdutide, maybe not mazdutide yet, but those two kind of more advanced drugs, everyone is doing a MASH trial as well, because glucagon has direct effect into the liver and has direct effect, meaning it does burn fat as well. So they're focused on obesity and MASH. But when you look at those two drugs, the glucose lowering effect is weak or non-existent. The reason is GLP-1 lowers while glucagon increases the glucose. So if it's well balanced, it balances it completely out, no effect. But if you have a much higher into the glucagon, then it will be lower on lowering the glucose levels. So it's a very interesting part how these two dual agonists with different mechanism of action can work together to enhance both of their efficacies. And that is the focus of us. The difference between us and the other, like GLP-1s, we are like semaglutide, tirzepatide, main difference, we're dual mechanism of action. Difference between other GLP-1 glucagon, we believe 3-to-1 balance highlights both of the mechanism of action to show the best from both.
Thank you, HH. Next question. What is the ownership structure of NeuroBo? How much stock does Dong own? And how does Dong support the business?
Right now, Donga ST owns about 60% of the company, the outstanding shares. And we are getting the research center, Donga ST Research Center, help on base science and lab work, because rather than spending so much money on opening a lab in the US, it's much more efficient to use a Korean lab plus those other scientists who developed both of these assets, 1241, 1726, from scratch. So they understand the most about these two products, and it was logical for us to ask them to provide us with the services. So that's one on the science side and the lab work. Then the next one would be the finance side. Ever since NeuroBo had the licensing deal with Donga ST to bring in these two metabolic assets, Donga ST have been providing 50% of the each raise. So they're fully backing us up with the science and with the finance so that we can progress with these two assets in clinical.
Thank you. Next question is, what is your burn rate? How much cash runway do you have? And will you need to raise capital in 2025? Would you like me to address this?
Yes, please.
So our burn rate is anywhere, it fluctuates anywhere from $5 to $7 million a quarter. The fluctuation is a result of running two separate clinical trials. One is winding down, DA 1241, and the other one is more or less in steady state, which is DA 1726 for obesity. We have disclosed in our Q3 results that we have cash into Q3 of 2025. And earlier on this call, HH discussed the outstanding warrants that are available based on a positive MAD result. MAD result is expected in Q1 of 2025. If that result is positive and if those warrants get exercised, that would bring in $20 million to the company, and that would fund us into 2026. HH, we don't have any other questions. I don't know whether you have other closing remarks.
Well, my closing remark would be just one thing to remember. We do have an obesity drug that had great results in preclinical, and we are running in-human trials as we speak, and we're running SAD and MAD at the same time, meaning the tolerability safety side looks great.